Hello, everyone, and welcome to today's presentation, Application of Brain Model Technology, presented by Dr. Alysson Muotri, Professor in the Department of Pediatrics, Cellular, and Molecular Medicine, and Director of the Stem Cell Program at the University of California, San Diego. My name is Robert Horton of Thermo Fisher Scientific, and I will be your moderator for today's presentation. Before I begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting any questions you may have at any time during this presentation. To do so, just click on the Ask a Question box located on the far left of your screen and type your question into the drop box that appears on the screen. We'll answer as many questions as we have time to at the end of the presentation. If you have any trouble seeing or hearing the presentation, please use the Ask a Question box to let us know of any technical difficulties. With that, please allow me to introduce today's speaker. Dr. Alysson Muotri earned his doctorate in genetics from the University of Sao Paulo in Brazil in 2001. He then moved to the Salk Institute as a Pew Latin American Fellow in 2002 for his postdoctoral training in the field of neuroscience and stem cell biology. Shortly thereafter, Dr. Muotri joined the School of Medicine at the University of California, San Diego, where he currently resides today. His research primarily focuses on modeling neurological diseases, such as autism spectrum disorders, using human-induced pluripotent stem cells. His lab has developed several techniques to culture human neurons and glia, from basic research to drug discovery platforms. He has received numerous awards, including the prestigious NIH Director's New Innovator Award, the Emerald Foundation Young Investigator Award, the Rock Star of Innovation from Connect NIH EUREKA Award, among many others. With that, I will now turn it over to Dr. Alysson Muotri.
All right. Thank you, Robert, and thank you, Thermo, for making this happen. So this is a nice set of speakers. I heard good things about previous speakers, so I'll try to keep it up the pace. And as Robert mentioned, my lab focuses on the applications of brain model technology, and what we mean is the ability to generate neurocells from induced pluripotent stem cells or human pluripotent embryonic stem cells. So one of the key things in my lab is that we focus on neurodevelopmental disorders, and we use those stem cells as a model system. One of the biggest limitations that we have in modeling neurodevelopmental disorders such as autism or even late onset diseases that have an embryonic origin is really the access to the embryonic human brain. So it's not ethical to use invasive methodologies to study the healthy human brain in uterus. Nobody would volunteer for that, but we rely on technologies such as ultrasound technology, things that don't have the high-level definition to really understand what happens at the level of single cells or even synapses. So because of that, we turn into these brain organoid models so we can recapitulate these very early stages of human neurodevelopment in the lab in controlled situations. And before I start, I want to mention that there are limitations as well, and it's nice to be upfront about among those limitations. So the media often call those mini brains in a dish, and I think the public perception is that we actually have a small human brain growing in the lab, and that's the wrong perception. So these brain organoids, they have severe limitations. For example, they are highly mature neurons. They are not vascularized. Not all cell types are represented. For example, microglia, who has a different embryonic origin, have to be transplanted back in some of the protocols. Even the ideal culture condition has been empirically defined, so there is no optimization of the median and factors. And finally, I think one of the biggest questions about this model, as well as any other in vitro model, is the translatability or the capacity to learn something from the human brain by just studying those neurons in a dish. So with all these limitations, we ask, can this technology be disruptive or is just incremental from what we previously had on the 2D monolayer cultures with these iPSCs? So over the years, my lab has been developing a novel protocol to create brain organoids, and these are patent brain organoids, so you can guide them to different brain regions using small molecules or factors. So most of the talk today is going to be about the cortex. And our protocol starts really with single cell level. So we dissociate iPSCs or human embryonic stem cells, and we keep them in single cell suspension for about 2 days. Only after that we induce, and it's a very short pulse of induction to the different brain regions that you want to target. In the case of cortex, we use dual SMAD inhibition, but we don't leave these factors over there. It's just like a short pulse. After that, we induce these neuroprogenitor cells to proliferate and really acquire a cell mass so we can follow the next step as neuronal differentiation, where these cells spontaneously will differentiate in the different subtype of neurons and glia cells that you expect during brain development. And again, I mean, we are not inducing them with factors. We are just relying on the natural programming, genetic programming of these cells, and they do that in a very reliable way. So, and then you enter the neuronal maturation phase, and this could last months. I mean, we can keep these organoids alive inside the lab for several years, actually, so it's quite robust. We do see some apoptosis or necrosis in the center of these organoids after a couple of months, but it's much later compared to previous protocols. And when we make a slice of one of these organoids, we have some kind of a structure. So this is a representation of a 4-month-old organoid. So you can clearly see that there is a ventricular zone, and this is where most of the progenitor cells reside, and they will differentiate into microglia cells that will send processes to the outer layers, where neuroprogenitor cells and glia progenitor cells will take advantage of those cables to migrate out to the periphery and start building what we call the cortical plate. As those neurons mature, we can start seeing the different subtypes of neurons appearing. And just to give you like a snapshot of what are the cell populations that we can get after 4 months, we have run single-cell RNA-seq, and this is just one of the representations of these maps. As you can see, in blue, most of the cells are glutamatergic neurons. By 4 months, you start to see that those blue cells start to divide between two populations. One is going to be upper cortical neurons. The other one is going to be more towards the lower cortical neurons. There is a population of progenitor cells here in purple that will continue to produce neurons and later on glia cells. At this stage, the percentage of glia cells is between 5 and 10%. It will grow up. I'll show you in a minute. And very interestingly, we also have a population of GABAergic inhibitory neurons that appear spontaneously from this protocol. So in a single unit, you can have both glutamatergic and GABAergic neurons. So you can use these kind of networks to test balances between excitations and inhibitions. So one can visualize the different cortical markers using staining, immunostainings, and I'm showing here the pattern of migration from the center of the progenitor cells, the ventricular zones, upper to the cortical layer. You can see at this stage you already have like some TBR2 transient cells as well as CTP2 positive cells in here. And I have two graphics here on the right. I just want to highlight two important things. One is the ratio between astrocytes and neurons. So this is percentage of cells over the course of 8 months. And at very early stages, 1 month, most of the cells that we have are neurons represented by the MAP2 positive cells in the black bars. And this population grows a little bit and then start to decrease. By the time it's decreasing, the percentage of astrocytes represented in the white bar by the GFAP positive cells, they start to increase. And just note that between 6 and 8 months, they kind of took over the entire organoid and becomes a 1-to-1 ratio between neurons and astrocytes. This is really what we'd expect for early stages of human neurodevelopment. So in the other graph, we have different subtype of cortical neurons, and we're representing here CTP2 and SATB2. And again, over the course of 8 months, what we see is that this population starts to stratify. In the beginning, most of the cells are CTP2 positive cells, but as we reach 4, 6 months of age, the number of SATB2 positive cells start to become similar. So again, I mean, this is a very nice protocol because it's dynamic. It's different from your regular 2D culture where you have most of the time the same subtype of neurons. And then we ask, I mean, can we quantify or can we match these organoids to normal human development? And this is a tricky question because, as I told you, we don't have a small brain. We do have pieces of cortex that are developing. So the comparison between organoids and the human brain is not straightforward. And in theory, one could not even compare. But we do it anyways. And one way to do that, it is by gene expression, global gene expression. So we take advantage of tissue banks like the BrainSpan, where we upload our global gene expression coming from iPSCs or different stages of organoids. Then we use other tools such as the CONTEST to really help us to make the match. And what we can see is that at the early stages, the iPSCs really match with embryonic stem cells, but as 1 month organoids already moved from that embryonic stages and are more similar to early, middle, fetal stages, and a 3 month organoid is already matching the global gene expression that is similar to late fetal stages or even a postnatal stage. So if you just look at gene expression, you might ask, how come like a 3 months organoid is already matching like a late fetal stage? And there are 2 explanations possible, at least. One is that perhaps the organoids are accelerated compared to normal human development. That's one explanation. The other explanation is the postmortem tissues are really not recapitulating exactly what happens because there are RNA degradation. We don't know exactly the age of these postmortem tissues, so there's lots of unknowns. But nonetheless, I mean, it's nice to see that there's a progression towards more mature neurons over time. So when my lab and other labs start to see that we can recreate or recapitulate some of these early stages of brain development, we look for diseases to model. And I think one of the early diseases that we decided to model was microcephaly. And I think the most striking example, it is the type of microcephaly that's caused by the Zika virus. And we took advantage of this model and exposed the brain organoids to the Zika virus. And that was a very important tool to prove causality between the circulating Zika virus in Brazil and the severe neurological defects and structural defects that we see in the brains of the fetus that were infected. So this is a published work, so I'll not give too much details, but just to say that the Zika virus were able to successfully penetrate into the ventricles of these organoids and kill transient progenitor cells. And as a consequence, what we have is a decrease in the number of certain subtype of neurons, such as CTP2 positive neurons, and these will reduce the thickness of the cortical plate. So that was a very nice proof of principle that how environmental factor could actually alter the structure of the human brain. So, but we can also take advantage of this model to test drugs. And this is one example. We have been using these brain organoids models to test drugs. This is one positive data that we get with the Zika virus. In the figure here, you can see that a mock-infected culture produced viable brain organoids, but in the presence of a very low amount of Zika virus. If you wait long enough, they would virtually kill all the cells. And we were able to find this drug called sofosbuvir, and these can protect the brain organoids from the Zika virus because it blocks the viral replication. So that was a very encouraging positive data using these models. So could repurpose these drugs for a possible new outbreak in one of these countries. But I told you in the beginning that we work with neurodevelopmental disorders such as autism, and those diseases are hard to find specific brain abnormalities. On the contrary, what we have at these stages or these diseases are networks defects. And we had no idea if these brain organoids could actually model neuronal networks. So this is one thing that my lab has been actively focusing to prove if these brain organoids would be a good model for that purpose. And to measure networks, what we did was to take advantage of multi-electrode arrays. These are printed electrodes in the bottom of wells where you can plate your brain organoid just on top of these electrodes, and you can record over time. The beauty of this technology is that it gives you network activity over the course of several weeks, if not a month. And it generates raster plots, such as this one represented on the top, where you can see that each channel, this is just 5 of the 64 channels that we have in here. And you can see that all the time when there is an event or an activity, we represent by a trace. So that's a spike event. If you have lots of spikes in a very short period of time, we call that a burst. And if you have lots of bursts across different channels, we call that synchrony. And our data has to be compared to something. So this is what we are plotting here is a mean firing rate in hertz, which is an overall measure of brain activity. And we plotted some of the work that was previously done using iPS-derived cells or mouse primary cultures. So I think you can appreciate that most of the work done so far, it is below the 5 hertz threshold. So how this compare to the brain? So a mouse brain would be around 18 hertz, and a primate brain would be around 20 hertz. So the 2D system is really not recapitulating that level of activity that one would expect for a more mature brain. And even organoids or previous protocols of organoids, such as this one here from Quadrate et al. From the Paola Lotta lab, they were able to keep the cells at up to 32 weeks and measure with multi-electrode arrays, but even though it doesn't reach a level of 5 hertz. So to our surprise, what we see is that at 16 hertz with our brain organoids model, you already see like a very high level of activity compared to most of the 2D monolayer cultures we had in the past. I'm plotting in this graphic an average of 8 organoids that was measured weekly over the course of 40 weeks. And you can see that after about 6 months, we see this peak of activity that will reach close to the 20 hertz, and after 40 weeks, it plateau. We don't see an increase in activity after 40 weeks. But nonetheless, the level of activity that we can get, it is already comparable to a more fully mature brain, which is amazing, and as far as we know, very unprecedented in tissue culture. And I can break down in numbers for you. So here I'm plotting different parameters that we can capture with these electrode arrays over the course of 10 months. And the number of spikes, for example, in the first graphic would move from 3,000 spikes per minute to a little bit over 300,000 spikes per minute. Again, this is something that we were very surprised to see that you can reach that level of maturation in vitro. The burst frequency will also increase. The interval between the bursts will decrease, suggesting that there is more connectivity coming from as we mature these organoids. I also mentioned that we have both excitatory and inhibitory neurons, and we did some pharmacology on top of these multi-electrode arrays to really prove that we have both excitation and inhibition. For example, when we treat these brain organoids with bicuculin, who can specifically block GABAergic activity, we see an increase in activity. Or if we treat with baclofen, mucinol, or TCX, These are drugs that will block glutamatergic synapses, and you abolish the activity coming from these organoids. So this is all very exciting. We can see a high level of activity. We can see inhibition, excitation in the same organoid. And if you have everything in a single unit, perhaps you can start measuring other things such as brain oscillations. And for those who are not familiar, brain oscillations are these neural oscillations that you can actually measure using an EEG cap. Because the human brain has so much activity, it will pass through the skull and it can detect it by these field electrodes. So you generate these oscillations. These oscillations are correlated with several human behaviors and diseases. I think the classical example is seizures. You can see in this graphic here that if there is a seizure activity, the EEG pattern or the shape of these waves will dramatically change. So they are really important, especially if you want to compare these organoids with the real activity that happens in the brain. So we start looking for the neural oscillations in these organoids. And the way we do that was by creating population spiking from our raster plots, as well as by filtering the data from the multi-electrode array and creating what we call local field potential, which is really the basis for these oscillations to arrive. And when they do arrive in the beginning, by 8 to 10 weeks, we see no oscillations. After that, the oscillations become clear, more complex by 6 months. And after that, they become quite irregular, less pathological. So this is a sign of complexity. You can visualize these oscillations in different formats. For example, I'm showing here population spikes and the voltage. These are the formats of these waves, the shape of these waves over time. As I mentioned, in 2 months, the oscillations are not there. By 4 months, you can see small peaks appearing. In 6 months, there is no questions that you have regular oscillatory activity in these brain organoids. And by 8 months, what we see is that those oscillations become quite irregular. And we see even what we call a rebound effect of these oscillations, where they seem to start to overlap. So we can quantify all of that. And this is the inter-event interval, and just how frequent the peaks of these waves become. And you can see that it increases over time. By 40 weeks, it's really high, 0.8, the maximum would be 1. And if you quantify the number of subpeaks on these oscillatory waves, we see the expected inverted U-shaped curve, suggesting that up to 25 weeks, what we have is just an increase in the frequency. But after that, we see that the subpeaks start to overlap because of that rebound effect. The only way for this complexity to emerge is if we move from a simple activity where neurons are connected to each other, moving to a more recurrent connectivity where synapses are being tried between different neurons, and finally reaching a complex activity that are more flexible, abundant, and recurrent connectivity among these neurons. So all of that is quite exciting, but can we actually compare that to anything related to the human brain? And then I come back to that question, can we compare brain organoids to the human brain? And so they are quite different structures, but similar to global gene expression. I mean, if you're comparing the number of genes that are expressed, yes, we can do that. It's not perfect, but we use the human development as a guideline. So we decided to do the same thing. Let's use the activity of the human brain as a guideline, and then compare exactly only the things that we can compare, and try to guess what kind of age we have from these organoids. One thing that I must say is that the adult EEG that I showed you before are quite complex. It's full of neural oscillations. There is no space between them. our organoid oscillations is much more similar to a preterm neonatal EEG. This is pieces of activity or times of transient activities followed by a quiescent time during them. This is the part where the brain is just silent, is quiescent. So when we are born, we have lots of this quiescent, silent activity compared to adults, but as we age, the distance between these activities shorten over time, and that's how we acquire these very complex EEG throughout life. So we decided that this would be some of the features from the EEG that we could perhaps compare with our organoids, and the way we decided to do that was by training a machine on several EEG features or parameters, such as quiescent time or frequency of the transient activities. And to do that, we were able to collect EEGs from neonates between 25 and 38 post-conception weeks. These are EEGs that doctors recorded when the babies are premature, just to make sure that the brain activity is healthy. So we take advantage of this material when we create a machine learning algorithm, and we teach an unsupervised algorithm to find what is the exact age of the person based on the EEG. And the machine does it quite well, much better than several doctors that I know, because the human eye is not optimized to distinguish between 25 and 26 weeks. But the machine can do it because there are qualitative parameters that we use to train this machine. After the machine was trained, we feed them with the organoid data and the multilateral array extracted data oscillations. And then we ask, can the machine matches the organoid features to the EEG features and predict the organoid age? And the data we have, it is a mixed feelings, let's put it this way. So in this graphic,
Well, hello everyone. This is Robert. Just it looks like Alyson's phone accidentally disconnected, so he's actually calling back in, and we'll be able to resume in just a second. I apologize for any technical delays here.
hello. Can
Hi,
you hear me now?
Yes.
Yeah,
Yes.
I think something happened. Yeah. All right.
Please continue.
I'll continue.
Thank you.
All right.
Thank you.
All right. So this is what we have as a prediction, and I'll give you one example of how this prediction was done. This is when we measure the inter-event interval between the different stages of the EEG or different ages. As I told you, the interval becomes shorter over time. And the graphic here represents the interval in our organoids. And by 40 weeks, it matches this red line here. That is something similar to a healthy postnatal human brain. So our organoids by 40 weeks matches some EEG features found on postnatal human brain, which is very exciting to us. So now that we have an ability to measure these oscillations, we start asking ourselves, what are the bases of these dynamics? How the cortex can generate per se these oscillations without any input and output? And that's a very fundamental question, and we decided to address that using genetic tools. For example, we use CRISPRs to knock down specific genes. One of the top targets that we have is MECP2. It is a gene involved in neurodevelopment. If you have mutations in MECP2, you might also have expressed diseases such as Rett syndrome, schizophrenia, or autism. So if you knock down MECP2 from these organoids, what we have is completely absence of these neural oscillations over time. They simply don't appear, suggesting that MECP2 is essential for the appearance of these network oscillations. But how come MECP2 can cause that? So we turn into single-cell analysis. We dissociate isogenic cells from the controls and the knockout organoids, and we run single-cell RNA-seq. The first thing that we observe is that there is a significant decrease between layer 5 and 6 neurons. The knockout organoids have about 50% decrease in those neurons. If we zoom in, what kind of neurons are present in layer 5 and 6, we actually find out that both CTP2 positive cells and SATB2 positive cells are reduced as a population in the MECP2 knockout organoids compared to controls. So this is just one example of one of the factors that can actually be responsible for the spontaneous emergence of these neural oscillations. We are testing other genes as well. We are also testing pharmacological treatments, different drugs that might inhibit these brain oscillations. So from a basic science perspective, this is very interesting, but we also want to help families with diseases. So we look for potential treatments for people with autism and other neurological disorders. So one of the bottlenecks we have is our ability to generate brain organoids in large scale. So there are many people that are hoping to create a more reproducible models that could be used for this purpose. So my lab has a small contribution in that. We recently published a paper where we can generate these brain organoids, cortical organoids, directly from a skin biopsy. So you just place a couple of fibroblasts directly coming from the skin biopsy in a well, and using a specific protocol, in several weeks, you can already have a brain organoid. That allows us to do a high throughput, so we could, in theory, generate as many organoids from as many different people as we want. And for conditions such as idiopathic autism, this is something that we really want to explore. Other environmental factors that might impact brain development on different genetic backgrounds might also benefit from this kind of technology. So we think that it needs further development, but it's a proof of principle that this can be done, and we're quite excited about that. So I'm finishing here. I'll highlight what you should take from this presentation. One is that the spontaneous development of neural networks on these cortical organoids, they resemble the brain biological program trajectory. So you don't need any kind of external stimulus to really follow the normal brain trajectory. This was something that was unclear if the human brain would require some kind of external stimulus, but at least in vitro, it does not. So we see delta high gamma phase amplitude that couples with the synchronous events that we see in the multi-electrode array, and this is a hallmark of interregional cortical communication. The only way for us to have that kind of rebound effects of the subpeaks of the neural oscillations is by having this kind of intracortical communication. I also told you that we can use this tool to model oscillations in a dish, and we have been looking for factors that are essential for these oscillations: MECP2, a gene implicated in several neurodevelopmental disorders, is a strong candidate, and likely do that by reducing the subtype of cortical neurons in layer 5 and 6. And finally, when we attempted to compare the brain organoids with features that are coming from a human brain, we use EEG from different post-conception weeks brains. And it seems like after 25 weeks, we have a very nice matched in vitro with the EEG features in vivo. So there are many people that are involved in this work. Most of this work was done by Kleber Trujillo and Priscila Negrais, 2 postdocs in the lab. All the neuro oscillations was done in collaboration with the Wojtek lab. The single cell analysis was in a collaboration with the Gene EO lab and the Lilia lab as well. I have a full disclosure. I have a company called Tismo that works on developing drugs for autism using this technology. My funding agencies are mainly the NIH and CERM, the California Institute for Regenerative. And I also would like to thank all the families that support our research with donations, with tissue, materials, and really to keep us motivated to find better treatments for all these patients. So this is what I have, and I'll be happy to take any comments and questions.
Great. Thank you, Alysson, for this very informative presentation. So with that, we will now begin the live Q&A portion of the webinar. If you have any questions you would like to ask, please do so now by clicking on the Ask a Question drop box located at the far left of your presentation window. Type in your question to the box that appears on your screen and click the Send button. And we will try to address as many questions as we can in the time frame remaining. So with that, Alysson, I'd like to get started with the first question. This one's a little bit more generic in nature. And I just was curious, a lot of the 3D presentations that I've seen over the last few years and months, there's really a common thing about having to move to implant your organoid or your 3D model to really mature them and to achieve that full maturity. What do you think is preventing? Oh, sorry. In your model, you've been able to achieve some very good responses. What do you think is preventing us from achieving this level of maturation in vitro for many of these other models?
Yeah, I think we spend lots of time on the protocol optimization, and there are very simple things that you can do to optimize your culture conditions, such as optimization of the media or the timing of the factors that we use. And I think it's really a combination of several factors that lead to a protocol that can reach that level of activity. In the past, we thought that we would need to transplant these organoids into a mouse brain or any other animals to achieve this kind of maturations, but it turns out that we don't. But even though, we see that there is a plateau in the activity by 9 months, and we think that this stage is the maximum that the cells can do per se, and at this stage, we very likely need to receive some kind of input and output. And we are working on ways to stimulate these organoids, hoping to have a better, even stronger maturation in vitro. But we are trying to stay away from the mouse models because at the end of the day, we want to do some high throughput studies to screen drugs or to model diseases. And the animal transplantation is really a bottleneck for us.
Mm-hmm. Great. Excellent. So we have a couple of questions from the audience. First from Seattle Children's Research Institute. Are your neurons in your organoids myelinated?
They are not. We are on the stage a little before where the myelination should happen. We could stimulate the glial progenitor cells to turn into oligoprogenitor cells, but we are not done that. These would require a second stimulation at later stages. So we are generating all this level of activity without myelinization.
Gotcha. And another question, what is the great advantage of using the NEUN antibody?
these are an antibody that would mark mature neurons. So we're just using to quantify the number of neurons that are present in our cultures. We do that using single-cell RNA-seq as well, but sometimes when you need a visualization at the protein level, we use the NEWN antibody.
There's also a couple of questions around how you prepare or isolate the initial cells for it. So I'll just read two, and maybe you can comment on both because they're closely related. Regarding your organoids from skin biopsies, how do they compare to other methods such as Lancaster and the Pascal method out there in terms of maturation and heterogeneity of the neuronal populations? And a related question is they were curious on how you dissociate your into single cells to start out your protocol.
Yeah. Right. So you guys can have all details because we place a manuscript that's under review into bioarchives. So all the details, how we dissociate the cells, how we prepare the cells, can be found in bioarchives. Just search for my name, and you're going to see the protocols available over there. But briefly, our protocol is different from, for example, Lancaster, because we patent them. We are focusing on the cortex. So we don't see other types of neurons. There is no hippocampal or cerebellar neurons in there. And as a consequence, we gain more reproducibility and more activity, because now all the resources for the neurons to form synapses are restricted to this subtype of neurons. So it really depends on your questions to use one protocol versus the other. So we are really focusing on cortical development. So that's why we are choosing to patent these organoids towards the cortex.
Gotcha. Excellent. A couple more questions here in the queue. Is there any need to incorporate vasculature into these to improve or speed up the maturation in vitro, or have you started to look into that?
Yeah, so everything that I told you is in the absence of any endothelial vascularization. And I think that's why they reach a size of about 0.5 centimeter, and they don't grow more than that. And I also mentioned that in the center, we start to see a little bit of necrosis, and that might be a result of that. So we and others are looking for ways to vascularize the system. But again, I mean, we don't want to go into an animal model because that poses lots of other questions. So instead, what we are trying to do is to create artificial vases that will penetrate these organoids so we can pump blood or nutrients throughout these vases, and hoping that they will survive longer, they will help on speed up maturation, and very likely these organoids will grow bigger.
Great. Another question from the audience is, how well do these models correlate with clinical treatment outcomes for existing drugs? How predictive are they? Yeah,
so that's a great question, and I don't have the answer yet. I can tell you that we are running now one big experiment that should prove just how good these models are. The experiment is the following. We are running a clinical trial for autism using a specific drug here at UCSD, and we're going to enroll about 30 kids and 30 controls. All the participants or the subjects will have their brain organoids generated in vitro. We're going to run the clinical trial in vitro just with their brain organoids, and we're going to use these EEG features to show potential for translatability. We should be able to inform the clinical trial on who's going to respond, who's not going to respond based on the in vitro data. If we have 100% match, we can say that the model is really good for that. So, but we don't expect that. We expect that the model might be informative, but in certain aspects. So that would be a very good scenario for us.
Absolutely. And along those lines, this is a closely related question. What are some of your current obstacles to transforming this technology to screening workflows to initiate some of that drug screening and toxicity studies you're talking about?
Yeah, so during these protocols, I think one of the major obstacles is automation. We grow these organoids, and it still requires lots of humans' hands to change media, to automate, to keep an eye on them, and this induces lots of human errors. So since these are long protocols, I told you that we can keep them for years. You can imagine the number of frustrated people that have their organoids after 6 months contaminated, and this is like not only a burden to the person, but the economics of the lab is also a factor. So I think automation is something critical, and we start to have some discussions with bioengineers along these lines. I mean, because it shouldn't be that difficult to create systems that will feed or take care of these organoids in a totally cell-autonomous way.
Great. Excellent. Another question here. Have you already tried to develop organoids using disease cells? And if yes, which diseases and what are your preliminary results? Are there differences in dimensions, developments, or conclusions?
Yeah, so we work with several neurodevelopmental disorders. There are some where the phenotypes are more dramatic than others. I'll give you an example. We published last year a paper in Cell Stem Cell describing the defects on organoids generated from people with Aicardi-Goutieres syndrome, a very dramatic neurological syndrome that causes loss of neurons. In that paper, we also described the potential mechanisms to explain the neurotoxicity, and we were able to repurpose a drug that was HIV retroviral drugs that could inhibit the loss of those neurons. These drugs now are in clinical trials, so the mouse model could not provide any insights into the neurological aspects of this disease, so it was really the human model that could do it. And we have several other diseases where we see phenotypes that correlates better or not so good to the disease. So the starting materials, most of the cells that we start in my lab are coming from skin fibroblasts or dental pulp cells. And we were able to successfully isolate those primary cells and reprogram, and they do just as fine compared to controls to generate organoids. Obviously, for example, in the case of Aicardi-Goutieres, we see a massive loss of neurons at very early stages. So you can see that the organoids are microcephalic from the beginning. So they don't achieve the same diameter as the controls.
Thank you. Quick question around the range of the heterogeneity of your models. For example, what is the percentage of organoids that pass QC to be available for analysis?
Yeah, so the QC is an important question. We have done some extensive quality control on these organoids to know what is a good organoid, what is a bad organoid. And we have compared organoids generated from different genetic backgrounds as well. And so there are some early QC steps that you can use to detect if your organoids will move forward into the differentiation protocol or not. Cell death and the amount of debris in a dish is some of the key things that we keep an eye on it, because we know once the differentiation is not going well, these organoids will not develop. They will be stuck sometime, and they might attach to each other. So they don't become like single individual organoids. They're just a clump of cells. So these are some of the QCs. Obviously, there are later QCs. Once you have these organoids in hand, you want to make sure that they do have the different cortical layers represented, or the activity level is what we expected with our basal line. So there are, along the way, there are several QCs that you can apply.
Thank you. And there's a couple of comments centered around the time it takes some of these, and if there's any ability to cryopreserve them or at some late stage and then bring them back into the screening campaign. Can you comment on that?
That's a great question. Yeah, no, that's absolutely a great question. We we keep an eye on that as well. So far, we have not been able to freeze them in thaw and continue with a viable organoid. So once you start the protocol, you have to keep them alive, and that's why some people have them going in the lab for years. The good news is that once you have a batch of these organoids, you don't make like one or two, you make hundreds, if not thousands of organoids at the same time. A single batch can give you enough to do like a proteomic, a gene expression, a single cell analysis, and your electrophysiology. So you don't have to generate several to get all the materials that you need. So this is the good news. But the bad news is really once you start, you have to keep them. We cannot stop in the middle. There is no way in the protocol that we can freeze it down and recover later. We are testing some factors like cryopreservant that it might work at early stages, but so far no success for us.
Sounds like the postdocs and your grad students don't have much opportunity for vacation sometimes then. That's a long commitment. It's interesting.
And that's why the lab has to be very social, and they have to like each other because they need to help each other.
Absolutely. Another question centered around how could you explain the different phenotype in AGS organoids compared to the normal brain development in patients?
It matches really well because in the patients you can see already using ultrasound that the brains of AGS patients are small, suggesting these massive neuronal death. And that's exactly what we see in the organoids. We see that neurons just die. And we think this is all because of these expression of endogenous retroelements that causes these immune activation of these organoids, and these are very neurotoxic because it acts through the interferon alpha. So we actually showed that if you block interferon alpha, these neurons will survive and can actually rescue the disease. The caveat here is that you have to treat at very early stages, so there is no neuronal loss. So the treatments that we propose have to start very early on. The clinical trial I mentioned was already like in a little bit older kids. So this is mostly for toxicity, and we don't expect a full rescue. What we expect is just to slow down the progression of the disease.
Good. And so with that, I'm just going to end with two future-looking questions for you. One of which is, I'm just kind of curious, because we have a lot of researchers online today that are new to organoids and 3D models. What keeps you up at night? What are some of the major challenges that are vital that you need to overcome in the coming months or years to really take your neuronal model to the next level?
Yeah, so this is a great question. I think these early stages of human development is such a black box, and we are so curious to know what happens. For example, the fact that we see that these oscillations are genetically programmed to appear and doesn't require any input from outside, this is amazing. I think this is something that I was not expecting to see, to be honest. I thought that an in vitro system will never reach to that level. Now that we know that, we want to know what causes that. What are the genetic information in these very early stages that makes these cortical organoids to oscillate? And furthermore, can we use external input after they're formed to really mature these brains to a level that would be closer to, for example, a 1-year-old or a 2-year-old? So that are the questions that we think we can answer in the following 2-3 years.
Great. And one final question. What are you most excited about in the organoid field? What are you out there seeing that you're just super excited about and will be the future of organoids?
I think, well, there is a couple of things, but one thing that we like to do in the lab is to use these organoids to study human brain evolution. My lab has a long interest on the question of what makes us humans from the perspective of evolution, and especially on higher cognitive status that the human brain has compared to other species. So we are taking advantage of these models to compare the human brain development between humans, chimps, and other primates. We have like the entire zoo in the lab that we can compare. But most exciting, I think, is the ability to take advantage of enzymes to edit the DNA and insert ancestral variants such as genes that are found in Neanderthals but not in modern humans to see what's the impact of those genetic variants in modern humans. So that will give us like a hint of what are the really genetic information that was essential for our modern human species to thrive, to develop really fancy technology, to be able to be here in a conference discussing this kind of data. We don't see monkeys doing that. We don't see chimpanzees doing that. It's only modern humans. So we are an outlier among the primates, and we want to know what happens at the genetic level that creates a brain that's so unique in the animal kingdom.
Great. Thank you again so much, Alysson. We really appreciate your time today and presenting on this model. I'm very excited to see your upcoming publications and your future work to see how your model progresses. So, before we go, please note that today's webinar will be available on demand for viewing through February 2019, so the next 3 months. So thank you for joining us, and once again, thank you, Alysson, for your time today. Wonderful presentation, and I hope everyone enjoys the rest of today's event. Thank you.
Thank you so much, Robert. Bye.